Sterilization and disinfection techniques. Sterilization is defined as a complete elimination or killing of all microorganisms, including their spores, and making the material object free from them. Types: physical methods and chemical methods. Physical method comprises of sunlight, drying, heat, filtration, radiation. Chemical methods consist of alcohols, aldehydes, dyes, halogens, phenol, surface active agents, metallic salts, and gases. Physical methods is shown in the form of a flow chart. Heat of two: dry heat and moist heat. Dry heat comprises red hot, flaming, incineration, hot air oven. Moist heat, temperature below 100 degree, at 100 degree, above 100 degree. Radiation, ionizing X-ray, gamma rays, and then non-ionizing infrared, ultraviolet. Filtration, depth filter, membrane filter, air filters are used for filtration. Then this is the most commonly and used method for sterilization in labs. There are different methods of physical sterilization. They are sunlight, drying, heat, filtration, radiation, as I have said earlier. Then we are looking into it in detail. First of all, sunlight. Sunlight possesses bactericidal activity because of the presence of UV rays and heat. It plays an important role in spontaneous sterilization. Drying. Moisture is essential for the growth of microorganisms, so we have to remove moisture content by the method of drying, but this is not much effective. Spores are unaffected by drying. Heat. Heat is most widely used method for sterilization. Dry heat sterilization and moist heat sterilization, which is a method which employs the steam under pressure. Dry heat sterilization, in this sterilization, control is the bacillus subtilis. The killing effect of dry heat is due to the coagulation and denaturation of protein, injury to cell wall and cell membrane, oxidative damage. Type of heat: red hot, inoculating loops, points of forceps and shears, spatulas, knives, needles. And then flaming for sterilization of scalpels, needles, mouths of culture tubes, slides and cover slips. Incineration for infected and contaminated clothes, cotton, wool, stoppers, animal carcasses, and pathological materials are incinerated in a machine known as incinerator. Hot air oven. It is a double-walled chamber. This is the most widely used method of sterilization using dry heat. The oven is usually heated by electricity. It has a fan or turbo blower, and the air is heated at a temperature of 160 degree for 2 hours, or 170 degree for 1 hour, or 180 degree for 20 minutes. The hot air oven's principle is as of coagulation and denaturation of proteins, oxidative damage, toxic effects of elevated levels of electrolytes. Advantages of hot air oven: it is used for sterilizing dry glassware such as test tubes, petri dishes, flasks, etc. It is also used to sterilize pharmaceutical products such as liquid paraffin, dustin powder, fats, and grease. Disadvantages of it is a slow process. It is not suitable for heat sensitive, that is the heat labile materials like plastic, rubber, media, and alcohol. Moist heat sterilization: it is done with the help of an autoclave. It is a kind of a steam heat sterilization. Principle is moist heat kills microorganisms by coagulating and denaturing their enzymes and structures of protein. If the temperature treatment for sterilization is below 100 degrees Celsius, then the process is known as the pasteurization. If it is at 100 degrees Celsius, the process is known as boiling or tintilization. Then if the temperature is above 100 degrees Celsius, it is known as autoclaving or pressure cooker. Then speaking of the pasteurization, it was developed by Louis Pasteur. This method is used for sterilizing fruit juices, beer, wine, milk, etc. Different types of pasteurizations are there. LTH, that is the low temperature holding method. It is used for the sterilization of milk, beer, fruit juices, are maintained at 62.8 degrees Celsius for 30 minutes. High temperature short time method, products are held at 72 degrees centigrade for 50 seconds. UHT flash method, milk can be treated at 141 degrees centigrade for 2 seconds. Then the temperature at 100 degree, boiling of water, tintilization, boiling. We all know what it mean. Then steaming at 100 degrees Celsius, tintilization was discovered by John Tintall. It is a sterilizing it is for sterilizing substances to kill the spores of bacteria. The process of exposure of materials to steam at 100 degree for 20 minutes for these 3 consecutive days is known as sterilization. It is also known as fractional sterilization or intermittent boiling. And temperature above 100 degrees Celsius, autoclaving pressure cooker used. Autoclave was first developed by Chamberlain. It is a kind of mechanism in which steam under pressure is employed. Then autoclaving is usually done at 121 degrees centigrade for 15 lbs pressure. Then advantages and disadvantages of autoclave is as of advantages. It is used for sterilizing a culture media, instruments, dressings, applicators, etc. Disadvantages is that the fats and oils cannot be sterilized by steam. Pressure cooker was invented by Dennis Pan. It is used for a small scale manner of sterilization. And sterilization control is Bacillus thermophilus. Filtration, different type of filters are being used: air filters, syringe filters, membrane filters, cinctured glass filters, asbestos filters, and candle filters.